Good morning, everyone. Hope you had a good evening, had a chance to digest all the amazing information discussions we had yesterday. So we have a full half day, two-thirds day scheduled today. We'll have two full sessions. The first one will be Interoperability of Biodiversity Measurements, chaired by Harris Lewin. And then session 4 will be chaired by Scott Edwards, delivering change, where we'll be trying to create a synthesis and think about actual next actionable steps, including the PNAS piece. We will be using the breakout groups to build towards that, as we did yesterday, and Bill will make a couple of comments about that right now. Thank you.
Thanks, Gene. It's nice that to recognize the importance of the work that's being done here, they made today International Biodiversity Day, which is kind of good credit for our work. So our plans are that in the breakout session today, the first breakout session, we're going to think about what are we actually concluding from this. And the way we'll do that is that you go to your groups, your session 3 groups on your back. Go to that. We'll ensure that there's only one group leader from the themed groups per table. So we'll please distribute yourselves. So if there's another person on your table that chaired one of the groups, then move to another one. And then the chairs will then talk about, they've been asked to produce a set of bullet points of their conclusions and describe those, and then think about the bullet points, because these are the conclusions of the meeting. So discuss those, improve those, etc. And then in the the second breakout session, we're going to think about what is it that we're really trying to say from this? What's our key message? And the way you can think about that is, what is the title of the paper? And what, if you like, is the stand fast? What's the one-line conclusion of what we really want to say? So we'll ask you to be doing that, and you might want to put some thoughts into that as well.
Please welcome Session Chair of Session 3, Harris Lewin.
That's my cue. Okay, so I think most of you know Pam Soltis was the original session chair. I'm sitting in for her because she couldn't catch a connection. Pam is the director of a very large NSF program called iDig Bio. So Pam actually knows a lot about this subject of interoperability, much more so than I do. I promised Pam that I would, to honor her and her contributions in this area, she sent me the introduction, and I will honor that introduction by reading it to you. And before I do that, I just want to say a couple of things. First, what a pleasure it is to be here and to meet so many of you and to hear the fantastic science yesterday and topics that were discussed. And it really has been a illuminating experience for me as a genomicist to meet all of you, many of you, for the first time. Okay, so just to remind you a little bit about the session. So I am going to give the first talk on standards as they relate to interoperability. And I'm going to use the Earth Biogenome Project as an example of how the community has set standards. And I think it's a very strong example that other disciplines and other groups represented here can adopt and use. Mark Blackster will talk more about the Earth Biogenome Project, really drill down into the details and talk about the GOAT system, Jet's Genomes on a Tree, which is truly an interoperable database that incorporates data from many sources. And you're going to hear a lot about the Darwin Tree of Life project from him, which is the largest contributor to the Earth Biogenome Project. Christina Grosinger is going to talk about examples of data interoperability using her honeybee as a model system. And Oshin, I didn't ask him how to say his last name, Makoda, close, Makoda, is going to talk about AI. So with Pam not being here, I'm going to read her statement and I'm going to superimpose my own introduction over that. And it really covers all of these points that Pam made. And so I'll read Pam's statement. Monitoring biodiversity uses a multitude of approaches, generates heterogeneous data, and spans spatial scales and levels of biological organization, from microbial diversity assessed via eDNA to ecosystem scales using remote sensing. How can we synthesize these heterogeneous data for a more comprehensive assessment? For a more comprehensive assessment of biodiversity and how it is changing through time. Other fields of biology have also faced the need for data interoperability and integration and have successfully met this challenge. For example, the Darwin Core Standard specifies a set of terms intended to facilitate the sharing of information about biological diversity by providing identifiers, labels, and definitions. Darwin Core is primarily based on taxa, their occurrence in nature as documented by observations, specimens, samples, and related information. It has been instrumental in aggregating data from natural history specimens and observations into regional and global resources, such as the Global Biodiversity Information Facility. But species occurrences are just one facet of biodiversity, and the diversity of measurements described in yesterday's session, as if Pam was here somehow, requires innovative approaches to interoperability. Our speakers today all represent fields and research programs that have successfully integrated diverse data, and we hope their examples will help prompt solutions to the larger challenge of integrating biodiversity data. In this session, we will also explore how AI applications can be used for biodiversity monitoring and how AI can perhaps contribute to data interoperability. And that's Pam's statement. Okay, so I'm going to talk about how we established and how we promulgate standards for biodiversity genomics using the Earth Biogenome Project as an example. Okay, and just a little bit of background, the Earth Biogenome Project actually started, at least the concept of the project began in 2015. And the four co-founders of this project are shown here, one of whom is Gene Robinson, who's sitting in the room, John Kress, who is the Undersecretary for Science at the Smithsonian Institute, Wanming Yang from BGI, and myself. And the Earth Biogenome Project did not arise as an opportunistic thing. Hey, we can sequence everything, why don't we do it? It arose because four of us were facing challenges in our own research where we needed more genomes, and we just didn't have enough genomes. And I'm an evolutionary geneticist. I work in mammals. Gene was working in honeybees, and John is a plant ecologist. And together, we sort of formulated this idea, and the first meetings actually occurred across the mall at the Smithsonian 10 years ago. And then over a period of 3 years, we were meeting regularly actually here in the Academy. So that picture is taken in the founder's room across the way in the other end of the wing, and I thought it would be useful to tie that together to where we are today. I mean, 10 years ago, I wouldn't have expected, how far we've come in this period, but we have made a lot of progress. And so the project took more than 2 years to formulate. We all had a different color hair and more of it, that's for sure. And I put this here just because I think it's important, because we're talking about having a paper, a prospective paper in PNAS, and this paper is really what launched the project. About 24 scientists from around the world, mostly genomicists from all over the world. In fact, there are four authors of this paper who were part of this group, including Pam, Scott Edwards, who's here, and Jean, and myself, and who's the fourth one? Let's see. Yeah, myself and Pam and Scott and Jean. That's four. And this paper, published in 2018, was the manifesto for the project. And it received a lot of attention, as you can see from the all-metric score. And so it is a great forum. And if we can produce something of equivalent value coming out of this meeting, I think it's going to be read. And it could serve as a framework for moving forward. And this was a very important advance. So this was published in 2018, and the project officially launched at the Wellcome Trust in London later that year in November. Whoops. And so what is the Earth Biogenome Project? The Earth Biogenome Project is a confederated network of networks that has the common goal of sequencing and annotating the genomes of all 1.8 million named species of eukaryotes in a period of 10 years. It was pretty ambitious. At the time, it's still ambitious, quite an ambitious project. And I just want to point out, again, 1.8 million, is only maybe 10% or a little bit more of the species that probably exist on Earth. So even this ambitious goal of sequencing 1.8 million species will only give us knowledge on just a fraction of the biodiversity that exists in nature. So we have to realize we're very early in this whole area and understanding, the genetic and genomic basis that underlies all of the ecosystems and important traits that we're all interested in preserving. And this project, the cost, estimated cost, I want to say something because I'm sure somebody's going to ask about it, is $4 billion over a 10-year period. And these are pretty rigorous cost estimates that we've done. We've just revised it down to about $4 billion from our original estimates. And the reduction in costs are due to the progress in technology, the cost reductions in DNA sequencing. And $4 billion may sound a lot, but we're talking about globally over 10 years. Okay? And what was the cost of the Human Genome Project? It was $3 billion, right, in 1990. $1990. That's $5.7 billion today. So we can sequence all known, if we can get our hands on them, we can sequence all known species for less than the cost of sequencing the human genome. And you say, well, what's the value of that? Well, if you look at the Battelle reports and reports about the human genome, at least a 65 to 1 return on investment. We don't even know. There are individual problems in agriculture that are $1 trillion problems. Okay. The return on investment of sequencing everything is going to be much higher. And you look at the cost of other big science projects like the Large Hadron Collider, right? 4.7 to build that. It took $13.2 billion to discover the Higgs boson. You can't eat the Higgs boson. Okay? I'm not trying to disrespect my colleagues here in the physical sciences, but it hasn't conserved a single species, and you can't eat it, but it does tell us a lot about the origins and evolution of the universe, and our project will tell us a lot about the origins and evolution of life on Earth. And so the goal then is to build a digital repository of life, and that will allow us to build a digital foundation for interoperable biodiversity data that all of biodiversity research can plug into. The project has grown to 59 affiliated projects based in 88 countries, involving now more than 2,000 scientists in 88 countries. So we've got getting close to half the countries in the world involved in this. And this is a sort of a schematic of the network of networks. The projects, the individual affiliated projects, are located on the outside. Now we're coalescing those under regional nodes. The first regional node has just been named. It's the European Reference Genome Alliance. Will become an umbrella project for all the projects in Europe. And we have beginning to receive applications from other broad geographical areas in the world who are interested in aggregating projects, and this is a very important advance for us. And then right there in the center, there's an elected executive council from the membership council, which draws from these affiliated projects. I chair the executive council, and the secretariat, for the global secretariat for the project, is now at Arizona State University, and that's supported by Arizona State University, the University of British Columbia, and the Wellcome Sanger Institute. And the role of the secretariat there are these incredibly important functions which we started from the very beginning of the project, that is to establish standards for the project, a solid governance structure for the project, as well as recommendations on policies and guidelines for such things as intellectual property. Okay. The project is committed to open access of data and compliance with the Convention on Biological Diversity and the Nagoya Protocols and on access and benefit sharing, and now the developing ideas around the use of digital sequence information, which is another important activity of the CBD. And the project is also committed, and we have a dedicated committee for this, and I'm not ashamed to say DEI here. So this is an important part of what we do. We're committed to diversity, equity, inclusion in the EBP community of sciences and with indigenous communities and local community, indigenous groups and local communities, indigenous peoples and local communities. So what is the progress of this project so far? So a lot of slides, a lot of data, but I'm only going to really show one slide here just to show you how quickly. If you look at the left side, that is growth in the number of sequenced species over the last few years. The last column is 2005, so the data are only for the first maybe 4 months or so. But as you can see on the left side there, we're now hitting about 4,000 genomes that have been produced by the Earth Biogenome Project affiliated projects. And what I want to call to your attention is the blue bars, because the blue bars represent chromosome scale assemblies that meet the metrics, that meet the standards of the that the Earth Biogenome has set for its assemblies. And if you look at the growth in the numbers, you can see that the growth in those numbers is really in the number of chromosome scale assemblies that are meeting the EBP standard. It's really critically important because it says that the affiliated projects of the EBP are adopting and using those standards, that we just didn't create standards that nobody can use. And if you look outside the EBP, if you look at the other reference quality genomes that are in the public domain in the INSDC databases, you will see many of those investigators have also adopted the EBP standards, even though they're not part of the EBP. So the standards are serving a very useful set of metrics for reference quality genomes. And I won't go into all of what those standards are, but I'm going to talk about them a little bit. And now, so these are the main points about interoperability that I want to make. And the contributions, what are the contributions then of the EBP to biodiversity data integration? First of all, as I just mentioned, standards for just about everything. You can go to the website, and I'll show you this in a moment, for standards for how to collect samples, standards for how to do DNA extractions across all eukaryotic taxa except for the protists, and we're working on the protists. And that's very important because they're a huge part of biodiversity that's still relatively uncharacterized and unsequenced. Standards for DNA sequencing, standards for genome assembly, standards for genome annotation and analysis, and IT and informatics. And all these are available on our website. And I'll just flip over so you can see, if you go, the reports here. These are all the reports for standards that are on our website. And you can see all the the standards that I just mentioned. But in addition, there are other reports around now. Governance documents are there, as well as documents related to intellectual property and other important foundational documents for the Earth Biogenome Project. I'll go back and say, what else have we done? Well, the group, the members have adopted that they will use standardized metadata, and the metadata will fit the Darwin Core Standard. So that's incredibly important in terms of interoperability and integration with other databases that use Darwin Core Standards. All of scripts and programs are put on GitHub, and the protocols for DNA extraction, for example, the way to do things, the way that will lead to very high quality genomes are put on protocols.io and shared for everyone. These high quality genomes, then they serve as anchor points. They serve as anchor points for anybody else that wants to use them because they're in the public domain. Project's members have agreed to put their assemblies into INSTC, which is the database of record. That's the International Nucleotide Sequencing Collaboration, and is the most really important repository, which includes GenBank, for DNA sequence information. Genomes on a Tree, Mark is going to talk about that some, so I won't talk about that, but that is the core information standard for the Earth Biogenome Project. And there's a lot of data and metadata that are included in there, and that's open to all, it's open to the public and is being widely used. And I'll let Mark tell you more about Genomes on a Tree. The thing is that it uses NCBI taxonomy, so this is something everybody can recognize. There's already sort of a filtering step there. We're not using 3 different types of taxonomy. We use one taxonomy in which everybody needs to conform to when they submit their assemblies. And other interoperable data sources, Mark, I don't know how many there are, at least a dozen, the Tree of Sex and many others, chromosome, DNA content, chromosome numbers in plants and animals and so on. There are all these databases that get incorporated into GOAT, and that this is the only place you can find all this information. Adherence to FAIR principles. INSDC, as I mentioned, is the repository for all the sequence. If you go to GenBank and you enter this project accession number, PNRJ, whatever, 33106, you can get every single, just by doing that, you can recover every single one of those 4,000 sequences that I mentioned that are in INSDC. So everything is organized under the umbrella of the Earth Biogenome Project, and then there are all the sub-projects and affiliated projects, so it's findable and accessible. Okay, and there's also a system developed, again, at Sanger, a brainchild of Mark, again, for standardized genome notes, which can automatically generate statistics about genome assemblies from the raw data without any human intervention. And now Mark and I have been working for the last year, almost a year now, on this universal genome node system, which is in collaboration with an outfit called Knowledge Futures, which uses PubPub, which will make the cost of publishing notes, individual genomes, basically free to the community, free. So if you think the cheapest you can get these published now is $100, and if you publish 1 million genome notes, just the cost of publishing the genome notes would be $100 million. Okay, now it's going to be free. And Mark, we're investing money from the Secretariat, and Wellcome Sanger is investing in this. So this is going to be a major innovation that will be implemented by the end of the year. Okay, mention this. Integration across biodiversity facets. So what are these annotated reference genomes good for? And this is really where we link to the rest of you in the community, in the biodiversity community. You can link genotype to phenotype, ecological trait databases, and other biodiversity measurements. You can put a link to the sequence in your database, or we can add links in GOAT eventually to your data. It strengthens eDNA. As we've heard yesterday, you can go into a soil sample and maybe only 50%, if you're lucky, you're going to be able to get a hit, so you can recognize what organism is there. This is going to contribute to resolving that issue. The more reference genomes we have, the better our eDNA surveys will be in terms of trying to describe an ecosystem. It will improve metagenomics and community composition studies and define evolutionary and conservation units. What is a species? Where are the hybrid zones? What are the units worth conserving? Are there really two species here, or is it only one? So this is very useful for answering those questions. Of course, those of you who are interested in quantitative traits and mapping other traits like adaptation to climate change, for example, this will support those efforts. Ecosystem level studies, understanding symbiosis. Mark is going to talk about that because they have a major project funded. Okay, my light's beginning to flash red. We're going to have a discussion about AI, but of course, if you have 1 million genomes, you can train your models on 1 million genomes. This is going to be the most useful data set ever created for really getting to the rules of life, because you're going to have genomes from all across eukaryotic life. Other disciplines, we have a collaborative global governance model. I heard a lot yesterday about the lack of standards. Well, this will help to address that. We have scalable protocols that we can now move from studying individual species in phase 1 is 10,000 genomes. We're going to phase 2 as soon as we hit 10,000, and we are implementing those protocols now to get us to be able to achieve 60 genomes a week or 37,000 genomes a year. That's the goal for phase 2. Robust open data, encouraging reuse and synthesis. This is going to be an eternal resource. We don't want to have to do this again. That's why we're doing reference quality genomes. Okay, so these data will be reused and reused and reused and reused for generations. Because this will be the infrastructure and the foundation for the future of biology. And an important message here is that early coordination is very important for the long-term success. So how can EBP help broaden interoperability, infrastructure for digital specimen integration with, for example, the iDigBio, any type of digitalized information, whether it be satellite information or images of museum collections. This is part of the complete digital record for an individual species. Interoperable ontologies across data types for traits, genomes, and environment. Training AI, which I mentioned, and importantly, capacity building and equity building through global access. We are spending a lot of time. Biodiversity is distributed throughout the world. We're not going to be able to access species everywhere, and so we're trying to bring the machines and technology to those places where the biodiversity exists in the world. And so we're trying to democratize the process of generating reference quality genomes through training and through delivery of technology. Mark, I don't think you're going to talk about that, today, but we do have some innovative ideas, genomes in a box and so on, that we could deliver in a crate anywhere around the world. Okay, and finally, interoperability needs and shared vision. This is our call to action. I think this is what we need as a community. Community of biodiversity researchers. We need to foster multimodal biodiversity platforms, of which this is one. Support technical infrastructure. That is clearly missing for a lot of the things I heard yesterday. There's not the equivalent of a Sanger Institute for many of the types of measurements that are being made. There are few standards, so there's a lot of this is missing, and I think this is what needs to go into our into our paper a call for community standards. Building bridges across the different disciplines that are important for biodiversity science, genomics, informatics, ecological monitoring, conservation, and policy. How to link all that up is critical, and empowering the next generation of sciences with tools with interoperable tools. So I thank you. I want to thank, this is not a one-man show. Those are our important committees, the elected executive council, the membership council representing all of the 59 projects. We have a committee on ethics, legal, and social issues, justice, equity, diversity, and inclusion, a governance committee, a nominations committee, a communications committee, and the International Science Committee, which has 5 subcommittees. Okay, this is all volunteer of experts around the world. 5 subcommittees dealing with sample collection and processing, sequencing, assembly, annotation, analysis, and informatics. And all those subcommittees are the ones who are responsible for getting those reports on the web. So thank you very much. 2,700 reference genomes and counting. And yeah, next time we meet, I think we're going to be very close to 10,000, our phase 1 target. So thanks very much. Our next speaker will be Mark Blackster, the head of the Tree of Life program at the Wellcome Sanger Institute. I don't think there's time. I think I went over a bit. Unless Anna wants to give me a few minutes. I think maybe 50. No, she doesn't. She wasn't sure if that included the 5 or...
Okay, so thank you, Harris, for the introduction, and thank you all for inviting me here. It's been a wonderful learning curve for me. I'm really enjoying this meeting. I'm going to talk about the work we're doing at the Wellcome Sanger Institute in the Tree of Life program to do with biodiversity genomics. So my background is I'm a zoologist, but most of my career was spent in a department of evolutionary biology, which has now renamed itself since I left as ecology and evolution. So I was embedded in a team of people who are actually working on evolutionary ecology questions all the time. And my question at every seminar that I went to was, have you sequenced it yet? Right? And for 25 years, the answer was no, because nobody had any money. But now we have the ability to sequence genomes at scale. And I jumped ship and moved to the Sanger Institute. And so Harris has introduced you to the Earth Biogenome Project. I want to talk about a smaller part of that, which is the Darwin Tree of Life Project. So shortly after that paper was published, a group of us gathered together in the Sanger Institute. And we all signed, I don't know what this is. This is a founding document. We all signed this founding document that said we would sequence all the eukaryotes in Britain and Ireland. So that's about... We have a good list of the species in Britain and Ireland because there have been taxonomists, natural historians, and everybody wandering around the islands and fishing off the shores for centuries. And so we have a list of about 72,000 eukaryotic species that have been caught, captured, seen, and named around these islands. 72,000 is a small part of 1.8 million. What's quite interesting about that 72,000 is that they actually cover about 4,500 taxonomic families. And since there are about 9,000 or 9,500 taxonomic families described on the planet, that's a good proportion of the taxonomic families are represented by species in Britain and Ireland. So we thought sequencing Britain and Ireland is something where we can test out procedures to run at scale, but also always thinking globally. So that was our motto, if you like, is sequence locally, think globally. Those of you who were involved in second wave feminism will remember that was also a motto of second wave feminism. That's absolutely where this comes from. We want to be good players in the environment, in the community, and to develop technologies and processes that are applicable globally. Okay, so lots of famous people signed this. I got to sign it as well, which is great. And about a year later, we started the Darwin Tree of Life project. So it's funded by the Wellcome Trust. The Wellcome Trust funded half of the human genome, or slightly more than half of the human genome the first time around. So this funding for the human genome that Harris mentioned. And they're best known as a funder for human health. What's really nice here is they have recognized that human health and human societal health is embedded in the ecosystems of this planet, and therefore sequencing the planet will deliver to human health goals, so perfectly within the Wellcome Trust's remit. So we sold Darwin Trust to the Darwin Tree of Life project to the Wellcome Trust as a project which would develop technologies for the whole world, would supply biodiversity genomics information for the whole world, but specifically answering questions about the ecology of Britain and Ireland and possibly Northern Europe. But it would also foster bioindustry and biotechnology. So we're going to sequencing species in which genes do interesting things, and those sequences might be of utility to humans. So our first goal was to sequence phylogenetic diversity. This was, it would have been easy just to sequence the mammals because we knew how to sequence mammals and we knew we could do that at scale. But how do you sequence bees, jellyfish, trees, mosses, protists? Just say, since Tree of Life started, it's nearly 6 years ago now, we started a number of other projects, and I've listed them here. The basic thing about these projects is they're all using this engine, this process we set up for the Darwin Tree of Life. And each of these projects is set up to generate a number of reference genomes to a particular goal. So, for example, the Aegis project, the second one on the list there, colleagues in Copenhagen are sequencing soil cores from ancient sediments. This is going back 1 million years, and getting DNA sequence from ancient sediments, and we're generating reference genomes against which to map those data. And their idea is to identify alleles at loci that will be of importance to agriculture and cropping systems from those ancient sediments. So those ancient sediments are from ecosystems which are different from ours, had different pathogen pressures, had different climatic regions, climatic pressures. So the idea is to use that to build a better agriculture today, etc., through the list. So to deliver all these, we built something we call the genome engine, and this is the genome engine. So as Harris said, the intention here was to be as explicit and open as possible, and to develop standards that we could promulgate across all our projects, but which also meshed with all our other partners. So the Sanger Institute is a sequencing center and really good at bioinformatics, but has no history in biodiversity. And so we collaborated with people in the biodiversity world, the museums, the botanical gardens, the Marine Biological Association, field study sites to collect specimens. And to do that, we used, as Harris pointed out, the Darwin Core essential data about each collection, such that the collections we made, the specimens we collected, they have photographs, they have locations, etc. And those are all available on GBIF. Okay, once the specimen's collected, we extract it. We had to develop all sorts of new extraction technologies. As I mentioned at breakfast, snails are full of slime. Slime to chemical extraction methods looks much like DNA. So if you extract DNA, or what you think is DNA, from a mollusk, what you get is a gram of slime and 0.00000, well, essentially no DNA. So we had to develop special extraction methods for lots of taxa. We've done that for most taxa now. We used long-read sequencing, mostly PacBio, Hi-Fi, but also ONT to generate the bulk of the data. We also use a technology called Hi-C, chromatin conformation capture sequencing, which allows us to link sequences together into the chromosomes of the original genome. And we also do RNA sequencing, transcriptome sequencing, so we have some data to help find gene models. Those data come off the machines and go on to what is called Tree of Life QC, which is a QC site which is publicly accessible. So you can go on Tree of Life QC now and see what we sequenced, what came off the machines yesterday, and check whether it's any good before any of us do. I mean, it is actually public before we see it. So the data are public and they're released to the public databases. We then do assemblies. Importantly, in the assembly, we also do curation. So this is a manual step, a largely manual step, of checking that the programs have got it right. The programs were all designed to work on humans. We're sequencing everything but humans. They make lots of human-specific errors, and so we have better there. Then all the data, the assembly goes to ENA. From ENA, it goes to Ensemble, where it gets annotated, and then it gets made public. So this is all very much open data. Currently, we've got 10,000 species collected, and we've released somewhere in the region of 2,600 species from across the tree of life. So the tree is a taxonomic tree, so it's not a phylogeny, it's just a taxonomic tree of life, with the protists at about 10 o'clock, the plants at about midday, ctenophores and jellyfish at about 1 o'clock, and then the deuterostomes, the sea urchins and vertebrates, and then all of the protostomes, and ending up with the butterflies. The red histogram bars are the ones that we sequenced and released. As you see, we've done a lot of butterflies and moths. This is partly for the project Psyche, which is sequencing lots and lots of butterflies and moths from Europe, but also because butterflies and moths are a real good testing ground for theories about genome evolution. And in all this, I said there's public data, and these places in circles here are where we make sure our data go public. And it's really important for us that these data are findable, are fair, interoperable, accessible, reusable. And so we're trying as hard as we can to make these data such. So Harris pointed, I've got some more slides, I'll leave them. Harris pointed out that there are 1.6 to 1.8 million eukaryotes on the planet. Any ecosystem you want to look at is going to have 3,000 or 4,000 species interacting in it. We've only generated sequences for 2,500 British and Irish species. So how are these data relevant to you? Well, first of all, they're the best genomes we can produce. So they are absolutely the best quality genomes out there. Compared to previous efforts at skimming genomes or doing small preliminary assemblies, these actually capture the totality of the genomes in a very complete way. So we can look at pieces of DNA which otherwise would have been lost. So I think if your species isn't on our list already, we can get it on our list, or if you want to sequence it, our technologies are available. We've published how to do it. Our estimate inside the Sanger Institute is it costs us about $1,500 for every gigabase of assembly. So a mammal costs about $4,500 to generate a chromosomal assembly, which compared with even a few years ago when it was running at $25,000 to $50,000 is pretty much amazing. The other thing is that we capture the entire genome rather than just a single marker like a barcode or a set of markers if you're using hybridization capture. And so you can ask also many more questions of these genomes and the species that they represent than you could of just a subset of the data. So suddenly you can ask questions about patterns of evolution of gene families across an ecosystem, or patterns of rates of evolution of chromosome change across an ecosystem. Okay, so I think I'll stop there. And we will leave it.
Okay. One or two questions for either of us. But you want me to bring him the mic? Oh, they have one there. Okay. Okay.
Thanks. So my question is, if I have generated recently a chromosome level assembly for a species, do you take that off your list of what needs to be done? And that's question one. And then question two is, if I have it, can I give it to you and pay you to sequence it?
So the answer to question one is yes. We won't. We're not in the business of sequencing anything twice. Resource is far too valuable for that. So as Harris mentioned, I didn't do. We have this database called Genomes on a Tree, goat.genomehubs.org. So Genomes on a Tree is a database where we aggregate all sorts of information about genomes, including whether or not they are being sequenced by any of the EBP projects, what state they are in, and status they're in that sequencing process. So just started, data generated in assembly. But we also aggregate all the data that's publicly available in INSDC. And so we would see that you had sequenced the genome and it would be off our list. We wouldn't sequence it again. In terms of paying us to sequence it, honestly, the administration costs would be higher than the cost of the sequencing, so we'd just sequence it. I mean, I'm not saying we can sequence every species you want to collect them here. There is if you like, an activation energy to get something sequenced, but absolutely we would sequence genomes of interest. We have four things get a genome into a species into our list for sequencing. One, if it's one of the family representatives for Darwin Tree of Life, that's a phylogenetic. Then if it's important, and that just means if it's important to you. If it's iconic, obviously we all like to sequence things, big fluffy things with big eyes because they get on the front covers of things, but that's really important for social engagement. And iconic could be a butterfly, it could be a salamander, it could be a plant. Or if they're interesting, and interesting just means you think there's something really interesting going on in this genome. So those 3 I's actually allow us to sequence a whole bunch of stuff off the main track.
Yeah, I'll just add one thing to that. If you have a species, you just go to GOAT, and you can enter your species, and you can see immediately if it's on somebody's list, if it's been sequenced, if it's in assembly, where it is, if somebody's collected it and targeted it. If it's on somebody's list, there's been some confusion about that. That doesn't mean that it is being sequenced. If somebody like Mark has a list of all the species in the UK and Ireland, but that doesn't mean he's collected 10,000 of the 60,000. So you have to use the list in a way that says, is it being targeted for sequencing or not? You'll find it on the list, but that doesn't mean he's going to sequence it in the next year or two. So there's that opportunity to interact with Mark. The second thing is all the projects, we use GOAT, and we've created a tool now for any project to check the lists of any other project. It's a duplication checker, and we send out reports regularly now to all of the affiliated projects that tell them what the duplications are on other people's list. And if you're ready to start something, whether it's already been done, it could be out in the public domain and not be one of the on anybody's list, but you have already done it, and you would be able to see that in the duplication checker. Yeah. One more. Go ahead, Kate.
Thanks, and congratulations on this incredible project. Could you talk a little bit about how you're navigating or contributing to the global agreements on DSI from the perspective of the Earth Biogenome Project?
Sure. You want me to?
Sure. So we just say in collecting the specimens, we abide by all local and international laws and agreements. So we are very strict on Nagoya ABS. And we usually go beyond standard ABS. So we, for example, if we access a specimen from Chile, we will try and set up a collaboration whereby the PhD student comes from Chile to work with us. In terms of DSI, Sanger and others from EBP are part of the discussion and have been very much promoting open use of the digital sequence information we produce to globally. So we started sequencing in Britain and Ireland, not just because it's well known, but because we have a right to. And we were able to say to the world, you may use these sequences without let or hindrance. So we can do that. And so we're also trying to be very good players in the open data space, such that we're releasing these data openly. And that's really important to us. Britain, as you may know, has a history of colonialism. And there are still many places we could legally sample from which Britain still has control over because of the colonial history, and we have not sampled from those areas explicitly because we want to be the best possible player. So that's why we say we sample from Britain and Ireland, not from the United Kingdom, which includes overseas territory.
Yeah. And I'll just say EBP is a member of the DSI network, and we have designated representatives from the EBP who attend the CBD meetings we're a member. We're an invited observer to the CBD meetings, and we have people who regularly attend those meetings now. Okay. I think we should probably move on to Christina's talk. Thank you. Our next speaker.
So thank you, Harris. This has been such a great workshop. I've learned so much. My talk is going to be split into 2 parts. So the first, we're going to talk about the factors that influence your biodiversity measurements, some of which you can predict, and so you can record this metadata as you're doing your experiment. But other things, as you'll see, really you can't get that information in real time, or you may not know what you need. And so that will bring us to the second part of this talk, which is how do we get the metadata metadata that we need to really understand and interpret our biodiversity measurements and take action with those. So in terms of the factors that we need to measure, so as we all know, the collection method that you use really influences the type of data that you get. So in the world that I am, which is bee biomonitoring, one of the standard methods is using these colored bee bowl traps. And so these are bowls that have colors that are attractive to bees because they resemble flowers. And they're filled with liquid. So when the bee comes near the bowl, they fall into the bowl, get collected in the liquid. And then every week or two, the scientists will come out and collect the insects, dry them, pin them, and identify them. So this is obviously a lethal trapping system, and it requires a lot of post-collection processing to be able to get the data that you need. But there are some limitations, right? So the sizes of the bee bowls really influences the capture rates. If you have these bowls in areas with lots of flowers, the bees are smart, so they go to the flowers and not to your bowls. So you may think that you have a very poor landscape, but actually you have a very good landscape. And of course, you're selecting for certain sizes of insects. Only the smaller ones will really be captured in this. And so one way to deal with some of these issues, specifically the lethal sampling and the time for the processing, is to leverage AI. And so we were very fortunate at Penn State. We're awarded funding from NSF to develop a new graduate training program that brings together entomologists, ecologists, engineers, and computer scientists to develop these non-lethal AI-enabled monitoring systems where we're taking some of these standard methods and changing them into being these automated systems. And so this is an example of an automated malaise trap, which we call Insect Eye. And our students are developing all sorts of different projects related to this. Some of the details of the sampling methods that may seem kind of minor or also can be very important in terms of determining what kind of data you get. And so my example from this comes from a study that was led by DJ McNeil when he was a postdoc in my lab. He's now an assistant professor at University of Kentucky. So we were in this study taking the data from Firefly Watch, which is a citizen science project where people are asked just to go outside their doors, count how many flashing lights they see from fireflies, and enter that data into the database. And so this has produced a really impressive data set over many years. And our goal was to take the eastern U.S. part of this data set and try to understand what are the major drivers of firefly abundance. And sort of unsurprisingly, major drivers are what time of year you're going out, right? Because they have a specific phonology, and how many minutes since sunset, right? Because the most abundant species are flashing right around dusk. And so once we could control for this, then we could look at all these other environmental factors like soils, right? Because the firefly larvae live in soils, the land use and land cover and climate, and also light pollution. And so we could see how these factors were affecting the fireflies, but only if we controlled for these very simple processes first. The species that we're working with are influenced by environmental factors, but at different scales. And so an example from this is work that was done by Melanie Kammerer when she was a graduate student in the lab, and she's now a research scientist at EcoData. So she was sampling using these bee bowls, spring and summer bee communities from 33 sites around the Finger Lakes region. And then she took environmental data and habitat data and integrated these through random forest models, which are machine learning-based statistical analyses. And what she found was that the spring bees were responding to factors at a 1 kilometer radius, and these are probably the spring blooming trees, but the summer bees were responding to factors at a much more local scale, so 100-meter radius, right? So again, this was kind of the opposite from what we would have predicted, but also something that we couldn't have necessarily generated the data for at the time that we were collecting these samples. There's also important seasonal lag effects. So for this, there's another example from a study from Melanie where she was using the USGS bee monitoring data set that Sam Drogi and his collaborators have generated. And again, using these random forest analyses, what she found was that the weather the previous year was a major driver, right? Which makes sense because how many babies the bees can have depends on the weather of the previous year. And then those are the bees that you'll see when you're sampling. But again, depending on which type of bee species and which community, it was different drivers that were the major underlying factors. Work that was done by Gabriella Quinlan, who's starting her own lab at University of North Carolina, Greensboro soon, looked at the effects of these environmental factors on plants. And so again, the bees and other pollinators are depending on plants for their resources, and the soil, the weather, the climate conditions are all influencing the amount of resources that the plants can produce. So these sort of indirect effects are very important. And then finally, when we think about plant pollinator communities, there are some pollinators that are very dependent on certain plant species. So even if you may have all the correct abiotic factors for your pollinators, if their plants are not there, then that will influence how they're behaving and how they're able to survive. So given those complexities, right, so how can we get the kind of metadata that we need to understand our biodiversity measurements? And so for this, I'm going to give you some examples from our Beescape project. This is a multi-institutional project that we've been running for about 7 years now. And sort of the goal for this was to help people see the world through the eyes of a bee, right? So how can we understand the resources and risks that a bee experiences in the landscape, both from a scientific perspective and then also from a managerial perspective. So first of all, you can obviously do very local environmental assessments, either by doing surveys or putting sensors out. This is an example from a study that we did with Wyman's, which is the largest wild blueberry producer in North America. They let us go into their fields and assess microclimate and microhabitat effects on bee visitation patterns and fruit production. This was a study that was led by Heather Grab, who's an assistant professor in Penn State, and also Vikas Khanna, who's a professor at University of Pittsburgh. And so we put out microsensors across these different fields. And as you can see, this is two examples from a cold plot and a warm plot. So in the same field, you can get very large differences in microclimate. But of course, we can't cover the entire field with these sensors. And so what Heather did was she used a machine learning approach to create a map across our field sites of these different temperature and humidity conditions, so that we could, again, precisely assess for these factors with our different measurements. We are fortunate to have data at a larger scale. So across the U.S., we have the USDA Cropland Data Layer, which gives us information on land use categories, and PRISM, which gives us information on climate data. But again, there's a lot of different kinds of data that you can extract from this, and not all of it is easily interpretable. So one of our goals was to develop ecologically relevant habitat quality scores that could be, again, used in our mapping systems. And so one example of this is work that was done by Maggie Douglas when she was a postdoc in the lab, and she's now a faculty member at Dickinson College. And she created the crop insecticide bee toxic load index, which works at a 30-meter resolution across the continental U.S. And to do this, she used that cropland data layer information from USDA. She used the USGS pesticide database, which tells you how much of which pesticides are being applied to different crops in different states. And then she integrated this with the EPA Ecotox database to know if those pesticides and those quantities hit a bee, how many bees would die. And again, all of this got integrated together and downscaled so that we can have this score at this 30-meter resolution. So this is great, but we want to be able to make these data available more broadly. And so we created this Beescape platform, which if you're bored of listening to me, you can go to it right now. And it's a mapping interface. So anywhere in the continental US, you can find your location, highlight your area of interest, and then get information back on land use, these habitat quality scores, the economic value of pollination services, the weather, certain weather indices from this year, the previous year, and the 10-year normal. And then we're also pulling in data from iNaturalist so you can see which flowering plants and pollinators are actually available right now. And so when we developed this, we were thinking about this for use by non-scientists, so beekeepers or growers or conservationists or landscape architects. But then we discovered that the scientific community was actually using this quite a bit. So people were going into this interface, painstakingly finding their sites and pulling out the habitat quality scores. So to help the scientific community, we've now put all of this code into an R-Shiny interface that, again, is very simple to use. It's called B-Shiny. And so this is where people are able to go into B-Shiny with their GPS locations or their raster files and then download all of these data. And so I think I've managed to thank all the key people who are involved in this, but I wanted to especially thank Sarah Gossley from the USDA ARS, who's been our collaborator with a lot of these projects. And if there's time, I'm happy to take some questions. I have a red light, but anyway.
Gene.
Thank you for the great talk. As we think about interoperability, were there any kinds of data types that were harder to integrate to get a single score?
So I think from the work that we were doing, so we worked with Eric Lonsdorf, who created a floral resource map, floral resource index. And in his case, this was a very simple way because it was an expert opinion analysis, right? So taking these land use categories, asking experts what they thought about the floral resources in spring, summer, and fall, and then giving a score to that. So that was sort of a one-step conversion of the land use into the index. These other things like the insecticide toxic load and the economic value, that required a lot more intermediate steps that required synthesizing information from these other data sources. And that was difficult because things were at different scales, and so they had to be adjusted and downscaled to the right thing. And so I think the scale issue is the greatest problem when you're trying to pull together these different data sets because they're just aggregating at different scales. But at least for Beescape right now, everything is based on the land use categories. And so we can start from that. And then every time that gets updated, we just recalculate the other scores.
So I'm thinking about bees, and I know you're thinking about bees, but I'm going to try to ask the question in a more generalizable way. So I was in the standardization subgroup yesterday, and we talked a lot about, what aspects of data collection and analysis should be standardized. And I'm just curious where you fall on the spectrum of how much we as a community and other research communities should be standardizing our field data collection methods. Like, given that in fireflies, whether you sample just after the sun sets or not, it has such a big impact on things. Like, yeah, you can account for that time of day that you sampled and include that as metadata, but how important do you think it is to actually try to come together and come up with standards in terms of when we sample fireflies and things like that?
So I think it depends really on who's doing the sampling, right? So I think for citizen science projects, you are so lucky that people are contributing to your work, right? So I think allowing to have as much flexibility with that as possible, and then adjusting for it on the back end would be it's sort of the way to go with that. With some of these more standardized approaches like the bee bowls, for example, I think at this point we do know enough about how those work that we can have something very standardized. I will say when Melanie was analyzing the bee monitoring lab data, her initial pass, she came and she said, I've got clear evidence of declines. And I was like, this is a great data set to see this. And it ended up just being a change in the size of the bowls, right? And so again, we control for that, we manage that, but that was very important that we knew about that to begin with. So I think the more you can standardize, the better, but I feel like a lot of work is just going to have to be done on the back end to integrate these different data sets and take what we can get from those.
Yeah, how well represented are bees in museum collections, and how are the trends from museum collections showing in terms of bee declines over time compared to more contemporary times that you've been working on this?
Yeah, so there have been a couple of different studies that have looked at this. So one of those, her first ones was from Sydney Cameron, where they analyzed, where are the bumblebees specifically in their historic range based on museum specimens, and then was doing a very large scale analysis of where are those species now. So you could very clearly see range contractions with certain species. There was a study, and I'm just looking at Hollis because you were an author on that, looking at the counts of species that have been identified from historical data versus proportionally counts now. And that's again examples, a lot of examples in the U.S. of species that have been previously identified, but now have not been identified, even though we have much higher sampling, both from citizen science and research things. So there are a couple, the historical samples have been used in a couple of different ways for that.
Okay, I think we're going to have to move on. So the last two talks really exemplify the diversity of data types that we are dealing with, from genome sequence to field data from bees. And now we have a talk on AI who's going to tell us how to make sense of all of this and extract useful information from it. So, Oshin?
I'm not sure if I can deliver on that, but we'll see what we can do. So, thanks everyone, and thanks especially to the organizers and also to everyone that I've had some really great conversations with. So it's been fantastic to be here. So my name is Oshin Make. I'm a computer scientist at the University of Edinburgh. Okay, so there's three pillars that are kind of the things that are responsible for the rapid recent advancements in AI. That's data, compute, and algorithms. Data really is the key component here, and I think the kind of transformative thing that is that's possible as a result of modern solutions, it's this integration of different data, so different modalities of data, whether that be images, text, audio. So these models are kind of ingesting all of this raw information and finding patterns in a way that really wasn't possible with traditional techniques. So my hope today is to kind of give a bit of a flavor for what's possible with these newer models in the context of biodiversity data. And then separately, I want to talk a bit at the very end about the role of evaluation, because data, yes, is important for training our models, but there's a really crucial piece here in the story, which is the data that we need to be able to use to kind of quantify performance of our models. Are our models fit for purpose? Are we actually measuring the right thing? Are we making the appropriate progress? And I think that that's a piece that requires a bit more attention. Okay, so yes, part of the one partial piece of maybe good news is that there is this amazing kind of growth of biodiversity data being shared online. I think Nick did an amazing job yesterday of kind of showing us all of the limitations and some of the kind of problems with that data. But to give one example, GBIF, the centralized repository, at the start of this year had about 3 billion observations in that in their repository. And so my title had this funny word multimodal in it. So multimodality is this idea that actually this data is not just one thing. It's not just a taxonomic entity at a location and time. It potentially also is other observational information like images. So we have this rich visual data that's being shared now by people online. But also there's additional metadata in the literature or also in the minds of experts. So we have this kind of rich, kind of multimodal kind of spectrum of data that's available to us. And, giving one example, iNaturalist, the Citizen Science Repository, who we work with quite a lot, they have on the order of 250 million observations, but each of those observations has an image. And also there, in many cases, is kind of detailed discussions or interactions or texts that are being shared by people online as they kind of discuss this data. So I'm going to talk a bit today about kind of two projects we've been working on that's trying to bring together multimodal data to enhance the ability of these algorithms to hopefully help people address questions related to biodiversity monitoring. Okay, and the first one is about species range mapping. So a kind of conventional species distribution model. And again, I've only got 10 minutes, so it's a bit of a caricature. As input, we take a set of sparse observations, for just one species. We put this through our model, and then we end up with some kind of dense prediction in the sense that we have a continuous quantity, maybe some kind of relative abundance, or we have some kind of binary representation like a presence or an absence. But we're taking sparse data and producing dense outputs. So in my group and along with collaborators, we've been trying to develop deep learning inspired joint species distribution models. So the kind of key idea here is that instead of thinking about building models per species individually, let's just integrate all of these sources together, and sources, I mean data from different species, to try and kind of build a mega model, for want of a better word, that shares information across species and fills the gaps that we have in certain species with data from other species. So we've been building models with kind of on the orders of tens of thousands of species inside of the one model, and then being able to produce outputs in the forms of kind of relatively spatially coarse range maps. But the key thing I think that's kind of an exciting direction in all this is then being able to leverage what modern AI techniques are really good at, which again is this idea we can leverage multiple modalities as source of data. So we can actually bring in text information, so we can train these models not just on in situ on the ground observations, but actually we can take in information from the literature, which might be descriptions of species ranges or their habitat preferences in completely unstructured text form. And so in a way that wasn't possible with more traditional statistical models, we can actually integrate this into a deep distribution model. And to give a flavor of the kind of capabilities that are now possible as a result of doing this, if we have a single observation, which is this white circle indicated by the arrow, these models can produce some kind of prediction where they think a species that is in that location might occur. But we can also condition those models on text. So we can say, here's a location in space, but also I have this extra information, which is, I know this species perhaps is found at higher altitude mountains, like the top right, or it's found in desert locations. So we're kind of bringing together these different pieces of information to then enhance our ability to control the predictions of these models. And this isn't just qualitative, we can actually quantitatively measure this. So we can take held out validation data where we kind of trust the range maps that are present in it, and then we can measure performance. So here, better performance would be a higher bar. And so a kind of traditional deep SDM without text information does this well. And if we add in this additional text information, actually we get a kind of pretty dramatic improvement in performance from kind of leveraging, again, this multimodal signal. And so we've been working with the team at iNaturalist to try and integrate these models into their pipelines. Specifically, we've been focusing on doing it to improve their computer vision performance, so the automatic identification of species, and using this as a kind of a geographical prior. But more recently, we've been trying to put together a species range data product, so trying to develop range maps for on the order of 100,000 species. There's still a lot to do, and I think they're very far from perfect at the moment, but this is kind of a first step in that direction. Okay, so switching gears, I alluded to the fact that earlier on that there's this rich repositories of not just observations, but we've also got images as well now in these data sets. What we can do right now is we can do things like search for species. I can search for species and locations at time. But this is just, in my mind, like the tip of the iceberg in terms of the kinds of visual questions people might want to ask from this hugely diverse set of images. So, for example, what if you wanted to find images of not just a California condor, but a California condor that's tagged and that has green 26 on its wing? Or, for example, maybe you're interested in hermit crabs and plastic waste on their shells, or you're interested in, mammal interactions with humans and so on. So there's this incredibly long tail of questions that people might want to ask. Right now, if you wanted to explore those repositories to look for those images, you'd have a pretty boring time of sitting down and just looking through hundreds of millions of images. So we've been developing AI techniques that allow us to query via language to then retrieve relevant images for your search. So think of it a bit like a Google image search, but actually we're enabling kind of a completely open-ended set of queries to be searched for. And a kind of key piece of this, it goes back to the very start, it's this question of evaluation. So we've been putting together benchmark data sets that are kind of representative, or hopefully representative, of a diverse set of visual searches that ecologists, for example, might want to do. And so with these data sets, then we can actually quantify and measure performance of vision language models and AI techniques on this specific task. And, we don't have to get into the details now, but we can kind of measure performance of a whole range of different tasks. And the takeaway is that these models actually are surprisingly good at being able to adhere to these queries that people have, but there's still a lot of improvement. And the kind of places where they don't work is, so in this example, I'm showing you a grid of images, and the case where it's green is the case where it's the correct answer and red is the wrong answer. So these are all images that the model thinks are relevant for this query. So it gets the species right, it gets tagged species, but it doesn't get the precise number. Or when you have more kind of complex, let's call it like kind of domain-specific language and so on, they're not particularly good at that at the moment. Okay, so to wrap up, or to kind of wrap up the last piece of this talk, there's been, amazing progress. I think AI techniques have been helpful along the way to do this. The only reason that that's been possible is because we've been able to create these standardized evaluation metrics, benchmarks, ways of quantifying performance. And that means that methods developers who are just not domain experts, I include myself in this, we don't have enough understanding of knowledge of the problems to make meaningful contributions on the ecology side. But what we can do is we can take synthesized versions of these problems and boil them down into things that we can then quantify and measure performance on ourselves. We've been able to do that on things like species identification from images, for audio, for detecting species in images. It's a kind of a very well-defined task. But when we get to more open-ended questions around spatial modeling, forecasting, we don't really have this kind of standardized benchmarking. So again, there is a lot of great work that people have done to try and measure performance on the models they developed, but there isn't really, like, in my mind, standardized frameworks that we can kind of give to people in other communities, methods developers, to allow them to actually make progress in parallel to the kind of domain experts. And I think this is kind of a really missing piece of this whole puzzle, is being able to kind of codify some of these challenges that you have into ways that other people can start working with them and make progress. Okay, so wrapping up, the takeaways here is that we have these amazingly powerful multimodal AI solutions. We can integrate diverse data sources. Evaluation is a big piece of that puzzle, because otherwise we don't know if those models are working, and we need to make progress on that. And that's definitely going to be an interdisciplinary endeavor. I think these two communities have to interact to be able to make that work. And big thanks to all my collaborators. I won't name everyone here, but there's a whole bunch of amazing people that have done a lot of the work on these projects with us. And with that, I will conclude.
Thank you, Ocean. Patrick.
Great talk. I was wondering if you or maybe other people have tried to include genomic data in deep SDMs. And could you imagine using the Earth Biogenome Project as one of the modes in your deep SDMs? And can you anticipate how much it might improve the predictions?
Short answer, it sounds totally plausible. I think I don't want to speculate too much as to what value that would provide to the model. So I'm sure there's people here who could answer that specific question more precisely than me. But I would say at a high level, when that data becomes available, there's no reason why that can't be integrated into these projects. And I think what's kind of the power of these models is really about finding connections between species. And so if we can find ways of kind of understanding their relationships and interactions, and then using that to kind of plug observational gaps. So if this genome data allows you to kind of do that, or whether it's, other functional trait data that allows you to do that, I think that's definitely very plausible.
Scott.
Very exciting. Is there a way, once you have your species distribution model, to sort of ask where's the weight of the evidence for this model? In other words, maybe different parts of the range have different kinds of evidence behind them. Is there a way to sort of quantify that?
Yeah, I think that's a great question. I mean, I'm not sure if this is going to directly answer your question. I think what hopefully this line of thinking will enable is kind of a scale of modeling of species that isn't really being done necessarily at the moment. So you're going to look at kind of whole compositions of tens of thousands of species on location. So I think that's going to give you a kind of a better picture on what's happening locally. So I don't think it necessarily, these are still kind of correlative models. They're not getting to underlying mechanisms or drivers, but I think they're going to hopefully give a level of insight that maybe isn't there already.
One more.
Yep. Yeah. So for your multi-species distribution modeling, so one challenge, especially for such kind of methods, are that the input occurrence data is very sparse for many species. And so how is your model able to account for those sparse sampling? And then for the predictions, how do you account for dispersal limitations and things like that, especially because a species can find suitable habitat somewhere else, but may not be able to disperse to those places. And
okay, great questions. Yeah, so the first part around the sparseness of the data, so natively the models just handle that sparsity. And so instead of trying to fit some huge, big, complicated model to a very small number of observations, we're fitting a big, complicated model to many observations across many different species. So I guess we're kind of relying that there's some fundamental kind of structure or relationships or interactions between those species that we're training on to kind of, again, fill in some of those gaps that are present in the observation data. We're still incredibly susceptible to biases in these data sets, and there's still a lot more that we need to do. And then I think on your second point about dispersal, I think this, again, is a whole missing piece of these models, which is a really exciting direction, is building in a bit more ecological, mechanistic understanding. Not like full hog and trying to like make that like the only thing in the model, but like just trying to distill down the core principles that maybe will provide a bit more regularization and constraining of these approaches. Thank you.